Okay, this is lesson 4, which is about evolutionary history and an organism's genome. This topic basically explored the usefulness of molecular studies in studying evolution and identity of individual organisms. Okay, firstly, we have to understand that the comparison of nucleic acid or other molecules can be used to deduce relatedness because of the difference, because of the passing of genes from parents to offspring, and also alteration or mutation of genes through time. This can be used to relate to relatedness or evolution of different organisms. You must also remember that different genes can evolve at different rates, even in the same evolutionary lineage. So you have to select which genes you need to use depending on the objective of your study. For example, if your study is about evolutionary events or relating evolutionary events or relatedness from organisms that diverged 100 years ago or millions years ago or billion years ago, you have to use DNA segment that change relatively slower than the others. Okay, these DNA segments are usually those genes that codes for important or critical function for the organism. Okay, so example of this kind of gene is the gene for the ribosomal RNA molecule. Okay, so our ribosomal RNA molecule is very important because it converts the genetic material into the usable protein molecule. Okay, another type of study for evolutionary history is the recent evolutionary events. For recent evolutionary events, you can discriminate group of people from the other, or a population of same species, different population in the same species, or you can use this for parentage or relatedness of same organisms. Okay, example of the segments that can be used for recent evolutionary events. For human, what we use is mitochondrial DNA if we are trying to deduce relatedness based on the maternal side, because as we know, traditionally mitochondrial DNA is associated with maternal inheritance, but recently there are studies that are saying that mitochondrial DNA can also be inherited in father, but most of the mitochondrial DNA still came from the mother side. Now, if you are trying to do evolutionary events or related recent evolutionary event studies from mother side, father side, so mixed genders, from mother, father, you can use the autosomal genes. So you can use the autosomal genes, so specifically the STR, or what we call the short tandem repeats. Now, there are also STRs in the Y chromosome, so if you want to study about the paternal inheritance, you can use the YSTR chromosome. Okay? Example of these kinds of studies. So for the evolutionary events from several hundred or million years ago, we can look at the tree of life that was presented by Carl Woese in presenting that there are actually 3 domains of life: the bacteria, archaea, and eukarya. As you can see, the divergence of the bacteria and the growth of archaea and eukarya happened million years ago. So even if it's million years ago, so they can still discriminate that event because they are using the rRNA molecule gene in comparing these organisms. Okay? Alright, another example for the other one naman, the recent evolutionary events, we can use it in discriminating group of people. For example, the origins of the Ainu people in Japan. The Ainu people are actually aboriginal inhabitants of Hokkaido in Japan. So they have very distinct culture and very distinct appearance. So their appearance actually they are hairy. They have curly hair, and they have deep set eyes, which is very unique from the usual Japanese people. So what they did in this study is to get samples of mitochondrial DNA and STRs from inhabitants of the neighboring countries of Japan. So they got samples from the East Asian countries, the North Asian countries, and of course the Southeast Asian countries. So they compare the mitochondrial DNA and the YSTRs of these groups of people, and they found out that the Ainu clustered with the Nivki. The Nivki are people living in an island that is actually a part of Russia. So as you can see in this stream, so it's very interesting. As you can see here, the yellow Asians clustered in one clade, and the brown Asian clustered in one clade, and the Ainu and Nivki are different clade. Okay, so as you can see here, no, the Ainu people is related not to the Asian, no, the not related to the yellow or the brown Asian, but to the Nivki people. But as you can see here, no, you can see here the branch is longer than the Ainu. So this means that the Nivki gene undergo different several genetic mutation already. So they are very distinct from the Ainu, but they shared a common ancestor. No, the Nivki and Ainu share the common ancestor than it is sharing with other Asian countries. Okay, so you can use such kind of studies. Okay, another usefulness of this study is about parentage and identification of individuals. One of the most interesting studies about this is the identification of the Romanovs. So we discussed, or we mentioned more Romanovs before in Bio 2. But remember, the Romanovs are the last monarchs, or czars, what they call czars in Russia, until 1917. Now, because of their attempt to keep the hemophilia, no, we discussed hemophilia in Bio 2. So because of their attempt to keep the hemophilia of the Crown Prince Alexei, no, they resorted to witchcraft and false priests. That's Rasputin, no, which add to the anger of the Russian people towards the royal family. Eventually, they were captured and killed in a firing squad. After the massacre, they attempted to dispose the family in an abandoned mine shaft, but they failed to do that, and the bodies of the royal family were disposed in nameless graves. Now, in 1917, a local geologist, Dr. Advodin, I think, was able to locate a mass grave containing the remains of the royal family and some of their servants. Now, in 1991, DNA analysis from the recovered remains were analyzed. When analyzed genetically, the DNA were analyzed and discovered and positively identified some members of the family. All the 5 of the 7 members were accounted, but 2 children were missing, 1 boy and 1 girl. So that's Alexei and one of the daughters of Tsarina and the Tsar. The identity of the missing princess was debated. So the Russian scientists were saying that the missing princess is Maria, but the US scientists are saying that it was Anastasia. So because of this debate, rather than bring closure to the nearly 70-year mystery of the fate of the Romanovs, the identification of only the 5 children of the 7 family continued to fuel speculation that somehow the two siblings were miraculously escaped, and made their way out of Russia. And since 1918, over 200 people were claiming to be one of the 5 Romanov children. Now, in summer of 2007, a group of amateur archaeologists discovered another mass grave, where they discovered 44 sets of bone and teeth, and they are actually wondering if this is the second grave where some members of the family, of the Romanov family, and the servants were disposed. So in the late 2007, the Russian government invited teams of scientists to conduct independent DNA testing of the remains from the second grave. So this is the result. No. Now this is the result. So the result showed that the two missing children of Tsar Nicholas and Alexandra was accounted in the second grave. So Alexei was positively identified and one female daughter. So which one is the second daughter or the daughter with Alexei, if it's Maria and Anastasia, cannot be fully identify in this study, because of course no mahirap, it's very difficult to identify the actual identity if it's Maria and Anastasia without the actual samples from the children. But this study were able to give us the full account of all the Romanov family, regardless if they were able or not to identify which is Maria and Anastasia. So they were able to give full account of all the members of the Romanov family, and can be concluded based on this study that none of the members of the Romanov family survived the execution in that morning of July 17, 1918. So that's one interesting studies here in molecular studies about relatedness of organisms based on molecular studies. And also, no, it's very popular now about Jessica Soho shows the parentage of those missing children. So those are also usefulness of molecular studies. Okay. Now in your learning guide, you will also see this figure. It's about some genes were constantly changing throughout time. So this means you can track back the time of divergence based on the amount of genetic material that were changed from the original. Okay, so this is about molecular clock, but molecular clock will be discussed in the next topic. Okay, next is about gene duplication and gene families. Now, gene duplication is very important in the evolution of organism, because gene duplication give way to the increasing of genes in the genome of an organism. It's very important for a simple organism to be complex, to have larger number of genes. Okay, so repeated gene duplication results in gene families, and duplicated genes can be traced back to a ancestral gene to the common ancestor. Okay, there are two types of homologous genes, the orthologous genes and the paralogous genes. When we say orthologous genes, these are genes that have single copy in the genome and can be said as homologous between species, and they can diverge only after speciation occurs. So meaning an ancestral gene were mutated that caused speciation. Okay? Now, paralogous genes are genes that result from gene duplication. They are found in more than one copy in a single genome. So they can diverge in clade that carries them and often evolve in new functions. Okay, to illustrate the difference between orthologous genes and paralogous genes, this is an illustration. No, the orthologous genes came from an ancestral gene that undergo mutation and speciation. So this mutation here, and then mutation here, and another mutation here in species A and species B. So this caused divergence or speciation of the organism. Okay, now for paralogous gene, a single gene were duplicated in the same genome. Okay, to give you an example, for example, the early globin gene undergone gene duplication that resulted into two chains, the alpha chain and the beta chain. Alpha chain and the beta chain can be found in the same chromosome or same gene material. So alpha chain and beta chain are paralogs. Now alpha chain, there is specific alpha chain for mouse and specific alpha chain for human and specific alpha chain for cattle. So the difference between alpha of mouse, human, and cattle are called orthologous genes. but no. So it might be mouse alpha, human alpha, cattle beta, human beta, or mouse beta. They are all homologous that can be traced back to a single globin gene. Alright? Now, orthologous genes are widespread and extend across many wildly varied species. For examples, human and mice diverged 65 billion years ago. And 99% of our genes are autologous to that of the mice. Okay? Now, another thing you have to remember, no, the gene number is not proportional to the complexity of the organism. For example, humans only have 4 times as many genes as yeast. So we are highly complex than the yeast. But our genes are just 4 times more than of the yeast. So it doesn't mean that there are more genes, it is more complex. Okay? So that's it. So let's we will be discussing the molecular clock in the next lesson.